So this is a video about the idea of the statistics of genetics, and we can produce these Punnett squares to follow the combinations. And what we need to do is there are two principles in genetics that are in use here, and we just need to talk about them. We've talked about them, but just need to set them out specifically. One of the principles is called the Law of Segregation, and this principle goes back to the time of Mendel, so about 150 years ago, and the birth of the field of genetics. And that is that the genotype is not blended, even though the phenotype appears blended. So we have the pink flower, and yet it's composed of a genotype of one red allele and one white allele. And when it's time to send on this genetic trait, either one or the other allele is sent on. Okay, so in this case, it's purple and white flowers, and you send on one or the other to an offspring. And that is that if you have multiple offspring, by chance, they might get this one or that one independently. So this occurs independently and randomly, and that's the second principle, and that is that the passing on of these traits, the purple allele or the white allele, is random. So a person could have 8 kids, and they all happen by chance only get that allele, that form, like that. So they all have the same allele from that parent. So that's the basis of this Punnett square, is this idea of making all the possible combinations from the two parents, and then because they segregate, only one or the other will go on to each offspring by chance, and then it's a by chance occurrence and an issue of statistical probability. So we can come up with these predictions, just like what's the chance when you flip a coin it'll be heads? The answer is 50%. So what's the chance probability that we get this combination? The answer is 25% in this breeding experiment. And so these numbers come up in the context of experiments where scientists do a breeding experiment, and then they have a population like a field of flowers, and they say how many of the different phenotypes are seen, and that can be also evidence for the type of alleles, dominant, recessive, or incomplete dominant alleles, like that. So the book talks through these in chapter 14, and it shows you this. You start out with these true breeding plants, so that means they must be homozygous. That's implied. They're homozygous. The F1 generation is going to be 100% heterozygote, and in this case, we have a dominant recessive relationship, and purple is dominant, and white is recessive. And so we have this, and then in this case, we have this surprising hidden carrier, effect, where the recessive allele can be there and is hidden. And then the F2 generation, we get this. Now, F2 is crossing together the F1s. And so we can make a Punnett square and look at the possible combinations in the F2 generation. And if you just look at that Punnett square, you see this. These are the possible combinations, each occurring at 25%. Because it's dominant recessive, that's going to be purple. That's going to be white, and these will all be purple. So we can talk about ratios, and we can talk about a 3-to-1 ratio for a dominant recessive cross, where 3 in F2, 75% of the plants are going to be predicted to be purple, but 25% will be white, based just on this chance. And then this actually shows the idea is these are numbers from an experiment. And so they took 1,000 plants, it looks like, or nearly 1,000 plants, and about 3/4 of them were purple and about a quarter were white. Would it be possible for them all F2 to be white in an experiment? The answer is yeah. It would be really, really rare, but these numbers can flex, and that makes genetics confusing too, because you can do an experiment over and the numbers can shift because of this probabilistic nature. So the book just is talking through these concepts and showing this 3-to-1 phenotype ratio. There's also this ratio. The genotype ratio is going to be 1 to 2 to 1. Heterozygote to homozygote to heterozygote to homozygote. So we got two ratios going on there with this cross. And the book just goes through this. There's that ratio of the phenotype and the ratio of the underlying genotype. Now, if this were incomplete dominance, right? In incomplete dominance, we would see this nothing hidden. So this ratio would be true here also. So for incomplete dominance, we'd have a thing where you'd have like purple and white, and then half the plants would be this light purple, we'll say, imagine that. We always think about this in introductory genetics. We always think it must be a grade, it must be like pink or a light purple. But it's true that it could be it's just different. It's an observable difference, and in some cases, that it could be some just other characteristic, like it could be yellow, and you're like, how does that work out? And the answer is, well, it's just kind of a surprise, but that's how that gene works, and we get down to the molecular level and see what its chemistry is. So here's the codominant story, right? And the ratios in the codominant story. The book is just trucking through them. The book does not make enough emphasis, of this, that it's just these three possible combinations. There are no other possible pairwise combination of alleles. You're either talking about a dominant recessive relationship, incomplete dominance, or codominance. The end. Then the book goes through a lot of this, just looking at the chromosomes during replication and meiosis 1 and meiosis 2, which is back in chapter 13. In talking and setting the stage up for 14 and 15. So we just need to be aware of that we're looking at chapters 14 and 15, and the stage was set in the book for that in chapter 13. And it's simple. We went through the chapter 13 meiosis very quickly because there are just a couple of simple concepts, and we just want to go back to them again. And that is that here's a human karyotype. Humans have their genome is 23 chromosomes, and one of these was inherited from mom and one from dad, and those are called homologous chromosomes. So homologous, they have similar genes, like gene for hemoglobin or insulin, but they may have different alleles. So homologous chromosomes, same genes, maybe different alleles of the genes. Again, we're just grabbing this concept because we've been dealing with it. And then after S phase, the DNA has been replicated, and the homologous chromosomes are pairs of sister chromatids, which are like photocopies. So we learned this is a tetraploid cell. It has actually 4 genomes, 2 from mom, 2 from dad. The 2 from mom are photocopies, identical replicated copies. And these are the 4 products. They could go on to, say, if it's a male, they could form 4 sperm. It's going to be haploid, so one of these is going to go to each sperm. Now, you'll see that 2 of the sperm will be identical, like that. So we can either have the trait coming on from dad's mom or dad's dad and sending on just 1 copy. And that's all that's going on here, is that there's a molecular basis for the law of segregation and the law of independent assortment, and it's based on the work that is done in meiosis that we learned about in chapter 13. Boom. It's really straightforward. And so that's that. And we can just look, here's another, just a different textbook, just following through on meiosis to make sure that you understand, to make that connection, the visual connection of those, like that. And so that's what's happening during meiosis, is that we're producing sperm and eggs in those combinations, and that's that. Then remember that there are other terms and concepts, so we're working with these, and then there are these additional terms and concepts that we're picking up as we go as well. And so that's that.